But really this is a story about where this product came from. Dr. Monica Alleman and Dr. John Madigan are faculty at the University of California. They study, among other things, corticosteroids and how they're transmitted to infant foals at birth, and how the dysregulation of these corticosteroids can affect foal behavior. And they do a lot of this work at a very large research facility, a very large foaling barn in Northern California, which is right down the road from Davis. It provides a great opportunity to work with a very large number of foals. The barn manager and the barn owners are very enthusiastic about participating in research. So it's a great opportunity for everybody involved. But as probably no veterinarian wants to hear going into January, there was an outbreak where all the foals that are being born started to develop very characteristic hemorrhagic, as Dr. Madigan referred to it, pipe stream diarrhea. These foals, around 48 hours of age, became very ill, and it seemed that it happened to every single foal that was being born. So very quickly, they sent samples to the UC Davis Veterinary Hospital, where they were tested for the sort of standard equine panel that they used there. They found that the cultures, the samples tested positive for C. difficile toxins A and B by both ELISA and quantitative PCR, whereas other things you might expect as causing diarrhea, like rotavirus, were negative. And so for those of you who may or may not be familiar, I think most of this audience is probably familiar, but just in case, C. difficile is a major nosocomial infection for humans, especially the elderly, and those who have been treated with antibiotics in a hospital. And in a very similar way, C. difficile infections can affect horses among various parts of life, especially if they've been recently treated with antibiotics. What's interesting about neonates is that many human infants are asymptomatic carriers of C. difficile. In fact, most pediatricians won't test infants for C. difficile simply because they know it's very likely that they'll test positive and they're totally asymptomatic. Or conversely, if infants do have diarrhea, it's very unlikely that they actually have a C. diff infection. Dr. Gary Magdesian, who's another faculty member at UC Davis, has been writing about C. difficile in horses and foals for a number of years, and he's pointed out that about 20% of foals that came into the UC Davis Vet Hospital also tested positive for C. difficile, even though they were asymptomatic. And so as most veterinarians would probably go to as a first-line drug, metronidazole was prescribed to treat the C. diff outbreak. Unfortunately, the organism didn't respond, and this outbreak continued. What they found is that after they cultured this organism, and again, this is an outbreak. Things are happening every single day, and more foals are being born. This is the beginning of the foaling season. And so the barn owner is thinking about how the rest of the foaling season is coming, and she has another 50 foals that are going to be coming. And they found that this organism was resistant to metronidazole. But again, Evolve's relationship with the university is very close. I was a postdoc there for many years, and I gave talks on campus, so I knew Dr. Madigan, and he knew that I had left to work at this company. And he started talking to some of the founders of the company who are faculty and asked them if there was anything that we could do to help. You know, metronidazole wasn't working, and what is the other strategy that you would go to? Is it, do you go to vancomycin or something? I mean, you really run out of strategies very quickly. And so we began to talk with him about this as a possibility to come up with a formulation from a probiotics perspective that could help with the outbreak. And so as a microbial ecologist, we know that C. difficile really acts well and can cause disease when there's a lack of proper, what I would call, ecosystem function, where you have a low functioning gut community that isn't perhaps, is very nicely shown in this infographic, it's probably not a community that's doing a good job of fermenting dietary carbohydrates into short-chain fatty acids. It's not deconjugating bile acids into the deconjugated forms. Both of those have a pretty strong effect on the growth of C. difficile and other Clostridium organisms. And again, this whole concept of antibiotic treatment, of breaking down the functions of the gut microbiome as a community, has been known for a very long time. If you want to study C. difficile in a mouse model, or for that matter salmonella, you have to treat the mice with streptomycin first to get a very reliable infection model. So all of this is pretty well described. And so from a microbial ecologist perspective, maybe not, it's sort of a simple answer, but in practice, is it as simple to solve? And a lot of the work that we were doing at UC Davis but in the academic realm was trying to understand how milk oligosaccharides, which are these complex fibers found in all mammalian milks, can actually shape the gut microbiome. So there are composition, their structure, their complexity are very different from the dietary carbohydrates that we consume later in life. For humans, where human milk contains about 15 grams per liter of milk oligosaccharides, equine milks are a bit lower, but the structural similarities are actually pretty striking. And typically, these complex oligosaccharides require a lot of different enzymes on the bacteria's perspective to break down the sugar and actually make it digestible. So there's very few bacteria that do a really good job of breaking down these carbohydrates. Conversely, if the carbohydrates are broken down inappropriately without sort of if they're broken down by bad actors, the individual monomers, the single subunits, can actually feed the growth of what we'll call the undesirable bacteria, things like E. coli, Clostridium. And so in the context of this outbreak of trying to come up with a solution for a barn that is really, facing some very dire straits, we started reading through the literature, and the results were not very encouraging. We found that a lot of the studies that have been done looking at C. difficile or C. perfringens outbreaks that have been published, they use B. animalis. They don't find very good results. They say that there's no effect on reducing the shedding or the symptoms of C. difficile or C. perfringens. There's a review that's really nice that actually wonders whether or not there's any clinical efficacy for probiotics. But when we started thinking about those particular trials and maybe a way that we could try and improve upon them and build upon the research that had already been done, we realized that these mare milk oligosaccharides, these equine milk oligosaccharides, were probably not being utilized by the organisms in these trials. And we know from our work in humans that if you pair an oligosacchαρide, a dietary oligosacchαρide, with the right bacterium, you can get a much stronger response of the gut microbiome than if you choose something that isn't able to ferment those oligosaccharides. So we worked with Daniela Barilli and a really nice postdoc in her lab who does a really good job characterizing complex mixtures of milk oligosaccharides, Sarjan Karav. And we actually characterized thoroughbred equine milk oligosaccharides for the first early lactation through the first week of life. And we found that a lot of these structures are very similar to what we find in human milk. So this 3100, these are two isomers of the same structure. These are also found in human milk, and they're present in very high concentrations compared to some of these other structures that are sort of fiddled around in here. But what's interesting is that the diversity of these structures, the same linkages are found in human milk. So we thought that we reasoned that we could apply the lessons that we learned in humans into foals and try and help during this outbreak. And what we knew is that some organisms grow very well in these structures and some of them do not. So Bifidobacterium longum subspecies infantis, or B. infantis, is an organism that we know very well. It grows very, very nicely on these structures. The two that I've highlighted here one of these is LNT and the other is LNNT. They're isomers, and without some additional mass spectrometry, I can't tell you which one's which. But we know that B. infantis grows very well on both of these. Organisms like B. animalis or Saccharomyces don't grow very well in these structures, so we don't think that they can have a strong effect in a nursing foal. And then this ability to grow we know from our work in humans, again, has a very significant impact on this ability of this organism to grow and persist in the gut. And so we set out to actually test this solution. So we chose this organism, Bifidobacterium longum subspecies infantis. This organism's genome was published about 10 years ago by David Mills, who is my mentor at UC Davis, and Carlito Labrilla and Bruce Sturman. They're sort of the milk microbiome connection group that we worked with at Davis for a long time. And we found that we could target the milk oligosaccharides with an organism like Bifidobacterium longum subspecies infantis, but we wanted something to compete against C. difficile for the broken down monomers, so we added a Lactobacillus plantarum, which has been really well studied. There's a recent paper in Science that actually used plantarum to reduce the incidence of sepsis in infants in India. And so these together, we added a lot more B. infantis of 300 billion CFUs per dose and 10 billion CFUs of L. plantarum together, and added those doses into foals that were being born in the facility every 12 hours, about twice a day, for 48 hours. So this picture is actually the day that we showed up at the barn. This is the only smile we got out of the barn owner for the whole visit. This was a very tense time. I mean, this is someone who's faced with the loss of her livelihood. And I told her, I said, I don't know that this is going to work. this is a really challenging problem, and the literature is sort of spotty on whether or not we can have an effect, but we're willing to try, and we know that it can't hurt. So we brought the stuff over that we had sort of assembled and formulated in the lab, and we went forward. And what we found is that before we intervened, 90% of the animals that were being born into the facility were developing C. difficile-associated diarrhea. 15% of those animals actually died of all the animals that were born, and only 10% were healthy. After we intervened, and that intervention included a lot of other things too, things like biosafety protocols, which bacteria don't really listen to signs about hand washing and foot baths as well as you would hope. But we found that after we intervened, we had a significant decrease in the incidence of diarrhea. All of the animals that we dosed from birth failed to develop C. difficile-associated diarrhea. There were some animals that were symptomatic when we showed up and then started receiving the probiotic. Their symptoms resolved in about 6 to 8 hours instead of 3 or 4 days like she had been seeing before. And then we followed these animals through the records that she was keeping too, and we found that later on the animals developed less diarrhea between 7 and 14 days, which you would probably expect to see this sort of the symptomatic full heat diarrhea that is nonspecific. So we found a decrease, but with a Fisher's exact test of all forms of diarrhea. Yeah. And so the end result was that of the 58 animals that were born at the facility over the foaling season, about half of them received GlycoGuard, which is the B. infantis L. plantarum mixture. Among the animals that received that mixture from birth, there were no cases of C. difficile-associated diarrhea. But the animals who didn't receive it from birth, if they, we asked her to withhold giving the product to them just to see if the outbreak was over and she didn't need to keep dosing them, and they actually still became ill. So for the whole season, these animals were getting ill. And we collected fecal samples throughout this time of the animals that we were providing this product to. Fecal samples that were collected, they were stored in a stabilization buffer, and then later we extracted the DNA in the lab. We also did some next-gen sequencing, as was described previously, and we found that when we used quantitative PCR targeting Bifidobacterium infantis, we could isolate that organism and quantify it even up to 14 days of age, so about over a week after the organism had been dosed last. And we also found that we could detect L. plantarum up to 7 days later. So these organisms are actually thriving and persisting, which is really unusual for a probiotic. Normally, in the clinical trials that I've done in humans and in pigs, it's very difficult even to re-isolate the organism that you're putting in. And we found here that we could isolate in very high numbers the organism that we had put in a week later. And then there's this slow decline over time. This is as the animals are moving out to pasture and start to consume more grass. We also, when we looked at the overall gut microbiome, that this change was associated with an increase of dietary fiber fermenters that are very well described. And so we think that some of the effects of colonization by B. infantis, again, looking at the physiology of Clostridium, they're very sensitive to changes in pH and concentrations of short-chain fatty acids, as well as deconjugated bile acids. And again, this is from a really nice review by Rob Britton and Vince Young. And we know that Bifidobacterium and Lactobacilli, during their growth, produce lactate and lower the surrounding pH mildly to cause, to change the sort of milieu that they exist in. We also know that B. infantis deconjugates bile acids very efficiently, and so it can contribute to these unfavorable conditions for C. difficile. There's also been some work in vitro that suggested that secretions from Bifidobacterium can help stabilize the tight junctions between epithelial cells. This is the target of C. difficile toxin, and so by trying to stabilize this barrier, we think that it can ameliorate some of the symptoms of C. difficile toxin. And so the conclusions, and perhaps the biggest limitation of this trial, is that it's not a randomized, double-blinded, placebo-controlled trial. There are a lot of confounding factors. The barn owner and the barn managers were the ones who were collecting samples for us and evaluating animals over time. We fully acknowledge that there is certainly an evidence, a potential for bias in that. But what we have done is actually follow up on a large randomized double-blinded placebo-controlled trial to actually validate these instances that we've observed. Because our conclusion is that a probiotic mixture did halt the symptoms of a C. difficile outbreak among all of the other infection control procedures that were implemented. And this showed a significant reduction in diarrhea in foals in the first 2 weeks of life. And really the lesson that, that we'd like to take home from this is that if you focus the development of probiotics onto specific times of life for specific applications or to help restore individual functions to the gut microbiome, you can have improved effects over sort of the non-specific use of probiotics. And so while there's many limitations to this study, I think it's still worth pursuing, and it's very interesting in the findings that we've seen. So I want to thank, so this is actually one of the foals that was sick, and we went back and took a picture with him later. So I want to thank Dr. Madigan and Dr. Alleman, who really brought us into a very tense situation, and I'm really glad that we were able to resolve this for the barn owner, as well as Shara Sheldon, who is a graduate student at UC Davis, and Andra Hutton, Claire Shaw, and Lindsay Contreras, who did a lot of the work for this in extracting DNA and the PCRs. So with that, I'm happy to take any questions.
Who's ready? I'll ask one while you're walking up there, and that is, I think it's actually a bit of a statement, but and that is, we were a part of that large, foaling trial that was, done there, and I think one of the important take-home messages is when that word all diarrhea is a critical thing, because we were looking at the incidence of foal heat diarrhea. And, not knowing anything about that, now it turned out we had 2 farms, as you know, and whilst we didn't have a high incidence of foal heat this year, we still learned a lot from that on those 2 things, and are still analyzing a lot of that data. But I think there's so much benefit in understanding what oligosaccharides the certain bacteria like. We're excited to move forward with something further in even next year.
Question for you. When you describe the reduction in diarrhea as far as outcome, can you clarify on what criteria was used and what observers used those criteria?
We define diarrhea, the C. difficile associated diarrhea, as the profuse, foul-smelling, hemorrhagic diarrhea. And so when that ended, we counted them as no longer being diarrheic. Runny stools were still diarrhea. Wet stools, formed stools were not diarrhea. So it's like sort of a 1 to 4. In humans, we would call it a Bristol stool scale.
Other questions? Do you want to add comments to what I just said about that project?
Sure, yeah.
Go ahead.
No, I think that we have some very interesting data that we will continue to analyze, and hopefully we can present it all in full, fully analyzed and fleshed out next year.
And one of the things that we saw was an increase in body weight in the foals, and it was blinded. So the people were doing it. It was a difficult study in that they wanted fecals and milk samples collected on a pretty regular basis, so it was tough on the farm. But they had amazing help, and in fact, the second farm stopped partway through because they didn't have enough help to complete it. But still, even though we didn't have high ends of diarrhea on that farm this year, there is a significant difference in body weight. Now we're looking at, has that held out through the yearling year? So because the farm weighs all their horses every month, so we're going to get a follow-up. It's exciting stuff.
Yeah.
Any other questions? Thanks you all for your attention and for sticking it out to the end, and don't forget the celebration.